Whatever.
Anyway, thank you very much for coming. And there's a couple of seats up here at the front if you want to come. There's another one here. Go ahead. You want to stand? Fine. Good.
If you want to keep the heckling position in the back there. All right, so thanks. I'm glad everybody came into town. I actually have never been in the Pence Gallery before. I always sort of look in from the outside, but I've never actually got in. So now I have a chance to go around, although I did not see any microbial art here, and I'm a little disappointed. Good point, too small. I wasn't sure what the sort of background of the audience was going to be, so anybody stop and ask questions anywhere along the way. If anything doesn't make sense, I don't have to get to the end. I just want to talk to you about sort of microbial studies, and what I'm going to do is do a little bit of what I'm interested in and the history behind these studies, and then talk a little bit about genomics of DNA. So I'm actually going to start off with you could say a story about a bird. It's not going to be about microbes, but hopefully you'll see why I'm going to tell this story. And then I'm going to give a little bit of introduction about microbiology and microorganisms and sort of the big picture of what we know about microbes around the planet, and then specifically talk for a while about how DNA analysis has helped revolutionize our understanding of microbes on the planet. So here's my story of the bird. So this nice bucolic setting, actually probably around the corner from this spot. This bird was seen. Anybody know what that is? It's an American robin. Very good. If you didn't know that, you could grab one of these custom books. Brought around, you can look through it. This is I have 4 of these, by the way, this version. I'm a birder, or an ex-birder, I guess you could say. So you grab this book. If you know something about the birds, you look it up in the book. So you take the book and you find the page of the American robin, and you can learn things about the bird and the appearance, and you can match it to the picture, and you can even get a map of the biogeography of the bird, and try and use this wonderful field guide that a lot of people have made over many years to help interpret what you saw. If you're a geek like me, you can have iBird on your iPhone. I took that picture like an hour and a half ago. So, and iBird has the American robin and has the field map. The map is a little different than the one in this book, subtle details. Okay, so let's go back to the scene. So, not that surprising. You see an American robin, they're pretty common, except for there's one problem. Where is this? It is in England. That is correct. If you recognize the types of cars or that they're parked on the wrong side of the road. It was in Grimsby. I don't know anything about Grimsby, never been there. It's got a grim name, but you'll see that the end of the story is pretty grim too in a second. So if you zoom out, you now see that this American robin is in the wrong place. And it is not where it's supposed to be on the map. It's supposed to be over in here. They're not supposed to be observed over there. And so if you knew about North American birds, you might be able to figure out what it was. If you didn't quite know and someone said, oh, look, there's a robin, you'd look up the robin in the Bird Book of Britain, zoom in on the robin page. That does not look like the same bird. And you'd get confused. It's a pretty distantly related bird. There's a picture of the robin there. And so you'd come back to this bird and you'd say, now that's really weird. That's unusual. That's not something that's supposed to happen. And because birders are sort of like me, they're kind of dorks. They'll tell all their friends. They'll post messages in online chat rooms. Before Twitter, they were all called Twitchers. I don't know if they're still called Twitchers with Twitter. It gets all confusing with the terminology here. But they'll share that information, and that's exactly what they did. I took some clip art, and all the people showed up at this house or this area where they found this bird because they were really excited. It was in the wrong place. These people had never seen an American robin in the wild before. Unfortunately, the story does not end there. A falcon also noticed the American robin and consumed it in front of about 200 or 300 birders watching this American robin. That's just a side point to my story. It was a sad ending for the bird, but it did get to travel. So it was a nice thing. So the reason I'm telling you this story is I have a dream as a microbiologist and a recovering birder. My dream is to have a field guide to the microbes, just like I have a field guide to the birds. And I want to be able to tell when something comes into a place that it is unusual and out of place, or out of time, or unusual genetically. I want to know, for example, for biodefense purposes, if tularemia shows up in the New York City subway system, is that supposed to be there or not? When a new strain of E. coli shows up in Germany, for example, is it supposed to be there? Is it out of place? Is it unusual in any way? When something happens in the human digestive tract, how can we figure out what's supposed to be there or what's not? We need something like this field guide to be able to just go look up where things are supposed to be, the types that they are, how to identify them, and figure out what's going on. So that's the prelude to what I'm going to tell you about in terms of microbes, but that's what, as a sort of ex-bird ecologist, I would like to be able to do with microorganisms. It turns out, unfortunately, it's very hard.
No.
It's a very sad story, I'm telling you. It'll get a little bit better, but not completely. There are many problems with this, but if you look at what should be in a field guide, what you want in a field guide is a lot of distinct information. You want the types of organisms, so like the different kinds of birds, the warblers, the finches, the sparrows, the owls, the hawks, etc. You want to know something about the function of those organisms, how they contribute to ecosystems, what they eat, like robins, and things like that. You want to know where they are in space and time. That's also called their biogeography, and how to identify them. And you'd like to know where they naturally occur, as well as where they don't naturally occur. For example, has anybody seen the flock of parrots flying around San Francisco?
Yeah.
It's completely awesome. They do not belong there, but they live there and they fly around, and you can hear them from like a mile away sometimes because they are also very, very loud. And so that's sort of the prelude, and what I'm going to do now is tell you just a little bit of background, sort of a bigger picture to introduce you to microbes, just for a few minutes before talking about DNA. So does that sort of background make sense about where we're going here? All right, so we don't have a field guide to microbes, in part because, as someone was saying before when I didn't see the microbial art here, they're small. That's how they're defined, of course, so I hope they're small because we define them as things that you can't see without the aid of some like microscope or some other device. And they weren't really well understood at all until people started to look in microscopes. Some people had inferred that there must be small living organisms in certain types of environments by the product of their activities, but they weren't really paid attention to until people started to look at them. And one of the pioneers in this was Van Leeuwenhoek, who made a little bunch of different microscopes. That's actually one of his microscopes. If you get Make magazine, you can get instructions that a friend of mine made on terms of making Van Leeuwenhoek's microscope. It's a really easy thing to do at home. You just need to melt basically a piece of glass and make a little lens to make his microscope. And he observed all sorts of organisms by looking at like his teeth scrapings. It's kind of gross, but looking in pond water in other places, and lots of diversity out there in terms of form. And if you just look in a microscope in a bunch of different places, you can see incredible numbers, sheer numbers of microbes when you look around. So in a gram of soil, there are estimated to be about 100 million microbial cells in that gram of soil. More cells of microbes on Earth than there are stars in the universe. So it's a huge number, more biomass than there are in plants and animals. Massive amount of organisms, large biomass, and if you look in people, there are estimated to be in any random individual about 10 times as many microbial cells as there are human cells. So we are more than just our own cells. We are sort of a colony of microorganisms. The diversity of microbes is amazing when you start to look at them. So if you look in a microscope at all sorts of different types of microorganisms, the diversity of just shapes, the diversity of form that they take is quite astonishing. You see these wonderful microbes that make sort of hard cases around them. These are great because they also fossilize. The microbes that make these hard shells, you have a decent fossil record of, and you can track them in the past. This is a multicellular bacteria, forms a fruiting body. This thing over here is a giant single-celled microorganism that a friend of mine picked up from the bottom of the ocean with the Alvin submarine. It's the size of your fist, and it's a single cell. There's all sorts of incredible diversity of microbes in terms of their form. These are these salt-tolerant microbes that their cells are square-shaped, and no one has yet figured out how an organism is able to make itself into a square shape like that. So there are a bunch of people trying to figure that out. But if you write a grant proposal to most funding agencies and you say you want to study the diversity of shapes of microbes, they don't really care. But the function of microbes is also incredibly diverse, and we've learned a lot about the functions that they carry out in addition to their diversity of form. So I'm just going to go through the sort of litany of types of microbes. So of course, most of the microbes people are familiar with are the ones that do damage, the ones that have been defined as pathogens in some way, and there are a litany of examples. I don't have to go into all of them here, but those are the ones that are best studied. The best, most funding opportunities are available for studying things that make plants and animals and people sick, and they're sort of well characterized, at least at a reasonable level. There are also a reasonable number of what are called good microbes. These are microbes that work in a beneficial association with some sort of host, also known as a mutualistic association. So we've seen, for example, with legumes are able to pull nitrogen out of the air via bacteria that live inside specialized organs in their roots called nodules. That is why you use legumes as rotation crops to bring nitrogen into the soil. They do this with a symbiosis with bacteria. There's all sorts of animals that depend upon microbes to digest their food. They help us digest our food a little bit, but they're not completely necessary for everything. Whereas ruminants, like cows, microbes are the engine that digests their food for them. There are some really bizarre microbes that I have studied in the bottom of the ocean. There are these giant tube worms, which if you've seen any of the sort of Discovery Channel videos about the Alvin submarine going to the bottom of the ocean, there are these giant tube worms. Some of them can be 5 or 6 feet tall. They're this long shell with a red plume that's sticking out of the shell that you see here. And inside of them, they have no mouth. They have no digestive system. They don't look like animals at all. And in fact, they function like plants, basically. They have an ecosystem of bacteria inside of them that fix carbon for them. In the bottom of the ocean, there's no light. Those bacteria use energy from chemical compounds like hydrogen sulfide to drive the reactions that turn carbon dioxide into sugars and vitamins and amino acids and other things for their host. So there are thousands of examples that have now been characterized of these mutualistic associations between various microbes and plants and animals, even between different kinds of microbes. So there's sort of lots that both the bad microbes and the good microbes have been studied in a lot of detail. The ones that I will talk about right at the end of the talk that people are just trying to get a handle on now are all the other microbes that we see in places, like all over our skin and in our mouth and in our gut, and we have no idea what they do. Sometimes we call them commensals, which implies that they don't really do anything, but we don't know that. Mostly they're just sort of unknown, this diversity surrounding the roots of organisms, on the leaves of plants, in the guts of animals, in the mouth, in their skin, on your walls, everywhere. We know very little about what they do. And I'll come back to that in a little bit when I talk about DNA. Now, sorry, Russell, I didn't get your picture in here in time. But one of the things that we study in my lab, Russell is a graduate student in my lab, is organisms that live in extreme environments, and microbes dominate a lot of these environments. So if you go to the tail end of the distribution spectrum of where organisms live, say the lowest pH, or the highest temperature, or the highest pressure, etc., it's usually microbes that are able to survive and thrive in those conditions. And they've been able to somehow alter their machinery such that they're able to do that. And for example, for temperature, which is really interesting, for many years people thought that nothing could live above about 60 or 70 degrees Celsius. There were theoretical arguments for why everything would fall apart. And it turns out the microbes didn't listen to those theoretical arguments. And what seems to be the limiting factor in where life can occur is whether or not water is liquid. If water boils, everything dies. You can't adapt to being in a boiling situation. But down here, in the bottom of the ocean, where there are hydrothermal vents, where there are cracks on the seafloor and hot water is coming out from where it's been interacting with lava or magma, the water is actually above 100 degrees Celsius because of the pressure. and it's still not boiling. And there are organisms that can live in that. There is an organism that has been found that its optimal growth temperature is 112 degrees Celsius. So, and that's because water does not boil when it's under higher pressure until above 115 at the place where this organism lives. So the availability of water that's not frozen or boiling appears to be sort of a major constraint, not the temperature per se. In Davis, you'd be shot if you didn't talk about the microbes that are associated with various sort of food production. We have learned how to make use of microbes that can take simple, relatively simple carbon compounds or complicated ones like different grains and turn them into something that we like, whether it's cheese or wine or beer or some product like that. And there's also, of course, lots of industries that are making use of microbes and the biochemical diversity that they have to do something interesting. If you look at sort of the suite of biochemical processes that are known in any organism on the planet, microbes occupy the vast majority of that full diversity of different reactions. They're able to manipulate. There are microbes that live on carbon monoxide, for example. I mean, they can do anything, basically, in manipulating chemical compounds, and if there's a little bit of energy to be scavenged there, they can live off of it. Microbes also play major roles in sort of running the global nutrient cycles like carbon and nitrogen and phosphorus and sulfur. They're not the only things that play important roles in this, but they play major roles in all of these processes. So we think microbes rule. That's the point of this whole sort of introduction. There's incredible diversity there. There's diversity of the form that I showed you, and there's diversity of this function. And I just sort of skimming just at the top of this diversity of function, which there's thousands and thousands and thousands of studies about. But what I haven't told you is the diversity of kinds of microbes. Like, what I was talking about with the field guide to the birds, what are the different, the warblers and the finches and the owls, etc. What are the different kinds of microbes? And the reason I haven't told you that yet is because we didn't understand really anything in detail about that until people started to look at the DNA of microorganisms. And so now I'm going to shift and tell you about sort of the history of studies where DNA was used to study microbial diversity in some way, as opposed to sort of the functional diversity. If you take a huge step back and you ask sort of what are the main types of questions that people ask, like when they want a field guide or when they're asking questions about microbes in the environment, you can sort of divide it into these two areas. Who is out there? That is, what are the kinds of microbes? And what are they doing? What are their functions? And trying to link those together is really important, so it's useful to know which microbes are doing which functions. Just like it would have been useful for the robin to know that the falcon ate robins, as opposed to nuts or something like that. I thought I would just talk a tiny bit about, before I get into DNA studies, that there are a lot of ways that you can study organisms without their DNA. I mean, I'm an obsessed DNA person, but don't want to exclude the lots of different ways that people study things. So if you're a plant or animal person, you can go collect things in the field. That's Vladimir Nabokov, by the way, who, in addition to being a writer, was the curator of Lepidoptery at the Harvard Museum of Comparative Zoology and was an obsessed butterfly collector. You can go do field studies in some way. You can use satellite imagery to look at the forests in the Amazon. You can set up a controlled experiment where you do what's called a quadrat in a site and you count what's in the quadrat. Poor kid doesn't know where the bird is, but you can observe large macroorganisms in the field, which is a really nice thing that you can't really do very well with microbes. And of course, you can go to interesting places when you do these things too. And you can do all of these with microbes, actually. So you can go out to a field site and collect. That's someone collecting from, I forget the name of it, but one of the geysers in Yellowstone National Park that's been studied for microorganisms. Did you go to that one when you were there? Or I can't remember the name of it. Craig Venter is the person who I used to work with, who sailed around the world sampling the oceans, collecting microbial samples. Satellite imagery is really useful for studying photosynthesis in microbes. You can do quadrats and look in a microscope and go send things into the bottom of the ocean and study the microbes there. This is really cool stuff, but it is very limited in a lot of ways. And one of the main reasons that it's limited is because of two real problems with microbes. First is the appearance. What microbes look like does not tell us what kinds they are very well. So whereas I can go look up birds, and in most cases I can figure out what they are just by what they look like. I mean, there's some that I'm really bad at certain parts of bird anatomy or something like that, where I wouldn't understand what the book is saying, but you can figure it out solely from their appearance in many cases. And much of the classification of these organisms was based upon appearance initially. We've backed it up by other studies in a lot of these organisms, but microbes, that just doesn't work very well for most of them. And in addition, there's enormous functional diversity of microbes, even among things that that are closely related. So even when they're really the same type, they can have different functions. And like, for example, one E. coli strain that makes you sick and one that doesn't. So I'll come back to that in a minute, but the sort of field studies of microbes are challenged because of these issues. And so for many years now, people have been using sequencing in a variety of ways as an indirect means to study microbes and to learn things about them. And that's what I'm going to tell you about in the next few minutes here. And what I'm going to do is take you through what you could call sort of 4 eras of sequencing in the study of microorganisms. And each one of them continues today. But I'm going to sort of show you the different approaches that people have used to use DNA sequencing to study microbes. So the first one, back up here, is what you could call using DNA to infer the relationships among microbes, to study what's called the tree of life. That's the evolutionary relationships among all organisms on the planet. And this has been an important thing to do among scientists, mostly since the time of Darwin. Darwin was sort of the first person to really convince scientists to look at the evolutionary history of organisms and to draw that evolutionary history by a tree diagram, like a family tree, but a tree of organism history as opposed to a tree of sort of familial history. There were many other people who then drew up their favorite trees of life after Darwin sort of convinced people that evolutionary history was important. This is one that's shown in lots of places by someone named Ernst Haeckel. And he had in his tree one main branch that corresponded to plants, one main branch that corresponded to single-celled organisms that have a nucleus. That's where the DNA is packaged inside a special membrane compartment. They're called protists. And one branch that corresponded to animals. And then he sort of threw everything else that could exist down at the bottom of the tree, and he had no idea what to do. Another tree that's cited a lot is what's called the Five Kingdom tree. You'll see this in a lot of textbooks by someone named Whitaker. And he had 3 main branches. In this case, he threw the protists sort of away from the main part of the tree and replaced them with fungi, had plants and animals. And then he had these protists in a big lump down here because he didn't know what to do with them. And then he drew bacteria on the tree, but they were icky and they didn't really know what to do with these other microbes. So he tried to fit everything together onto this single tree, showing the relationships of all organisms. But admittedly, people really didn't know what to do with most of the microbes because of some of these problems that I was saying with appearance and not knowing sort of what to do. And that all changed mostly with one person, this person. So this guy, Carl Woese at the University of Illinois, was sort of frustrated with the inability for people to include microbes accurately in these trees. And he realized that there was a way to do this by looking at DNA of organisms. And so I assume most people here know, but I'm going to just do the little DNA tutorial for a second, just in case. So DNA is the material of heredity. The sum total of all the DNA in an organism is what we call the genome. And DNA comes in these long strings of repeated subunit chemicals. It's like a long chain. And the chemistry doesn't really matter here, but the chain each component comes in 4 different flavors. And those chemicals are abbreviated A, C, T, and G. And the order of those chemicals, A, C versus A, C, G, 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 matters. And that's what encodes the instructions for most of the features of an organism. And that's what we call the sequence. The order of those chemicals is the sequence of DNA for an organism. And with various chemical methods, many of them used to be very painful, and now we can get a $800,000 machine that can read the sequence for us and spit out billions of letters. When I was an undergraduate, I spent a year and a half reading 1,400 letters, and I got a paper out of it. Now we can read, 100 billion in a few days. So the technology has changed, but the point is the same. You want to read that order of letters inside the DNA for a particular organism. From an evolutionary point of view, what Woese realized was that we could line up corresponding regions of the genome of one organism and compare them to other organisms in the same way that people line up skeletons from, say, different vertebrates. So you can take a particular region of the genome that codes for the instruction, say, to make the machine that translates RNA into proteins. All organisms have basically the same machine in them. All organisms that have cells have this machine called the ribosome. And the exact letters, the order of letters inside the DNA for those organisms that codes for that machine is slightly different between different taxa. The more closely related organisms are, the more similar the letters are. The more distantly related organisms are, the more different the letters are. And so what Woese, well actually someone named Emil Zuckerkandl and Linus Pauling first realized this, and no one applied it to microbes, and then Woese was the first person to really do this with microorganisms. Realized he could do this sort of comparative sequence analysis in the same way people did comparative morphological analysis. And he did this, he gathered a bunch of microorganisms that he could grow in the lab, read in a very painstaking way some of the strings of letters inside their this machine, the ribosome, that they all had, and he lined them up with each other, and he built an evolutionary tree. And what he got flabbergasted him and everyone else at the time. What he found, this is a sort of star-like diagram of the evolutionary tree that he found. What he found were three main groups of organisms on the planet. The eukaryotes, those are all the organisms that have DNA packaged inside this nucleus. That's what eukaryotes mean. That includes plants, animals, fungi, and all these single-celled protists that Haeckel and others had all put together onto the main part of the tree. The normal bacteria, that includes things like E. coli, and anthrax, and Mycobacterium tuberculosis, and Vibrio cholera, all the things that people had previously sort of known to be called bacteria. And then a third group of organisms that nobody knew about, that they did not have their DNA packaged into this special thing called the nucleus. All of these organisms had been grouped together previously and called prokaryotes, organisms without a nucleus. And it turns out that they were two distinct forms of life, completely missed by any other analyses. And so Woese had opened up a whole new window into microbial diversity by being the first person to place microorganisms onto this tree and understand the different forms of life that are out there. Now this is important for lots of reasons. The biology of these organisms is different than the biology of those organisms.
When was this?
1977 was the two papers that came out that Woese wrote about this. And a lot of people went like this when they saw the papers because they had sort of data that all had suggested this previously, but he was the first person to figure this out. One of the cool things about this third form of life at the time, and sort of even now, is many of them were extremophiles, living and thriving in extreme conditions, such as boiling hot springs, really salty places, pH of 0. So they're organisms that basically live in sulfuric acid, boiling sulfuric acid. I guess they like it. And so this opened up a whole new window into microbial life and allowed people for the first time to accurately classify microorganisms. And even though he didn't do everything after that, using this approach led to massive restructuring of what we know about microbial life. There were organisms, for example, that rickettsias, which cause things like Rocky Mountain spotted fever, and chlamydias, which you can guess what they cause, were grouped together into one group. They are from different phyla of bacteria. The reason antibiotics don't work the same on both of them is that they're not even remotely closely related. And that was discovered by these types of analyses. And there are like 2,000 papers where people had completely moved organisms into different parts of the tree, changing what we would do about them based upon this type of analysis. and you know, it sort of revolutionized what we thought about microbes. If you build one of these trees, for example, these are the only parts where the things you're familiar with mostly are, the plants and animals and fungi. The rest are microorganisms. And so most of evolutionary diversity is microorganisms. Side story, which is still sort of controversial, is that this third group, the archaea, seem to actually be more closely related to us, to eukaryotes, than either is to bacteria. So it's made them sort of a model system for a variety of studies.
Did you say in recent paper in Nature there is another group? Could you tell us a little bit about it?
Another group?
Yeah. Yeah.
Oh, you mean my paper wasn't in Nature? Oh, you mean the virus paper too? I will come to that at the end, right? In a couple of minutes, if you can wait a second.
Right.
Because that required a different approach than this. So Woese went and he characterized organisms he could grow in the laboratory. And that was not sufficient for characterizing all of life. And if you can wait, I'll come back to it in a second. So the problem with Woese revolutionized our understanding, but there was one big problem with what he did. What he focused on were organisms that people could grow in the lab. And he could extract large amounts of material from them to do this sequencing reaction, to read the parts of their components. Unfortunately, that's a tiny fraction of the diversity of life. So there's this thing called the GPA. I'm sorry for the students here, but don't get scared. It's the Great Plate Count Anomaly, not the normal GPA. And if you go to an environment and you take a sample and you split it into two, and you grow the things in half the sample, try and culture them, and you look in a microscope in the other half of the sample, and you count then what you see or what you're able to grow, in every case, there's more that you can see than you can grow. In many cases, there are orders of magnitude more cells that you can see than the types of things that you can grow in the laboratory. People have known about this for about 100 years now and realized that the stuff we were able to grow in the laboratory was not everything that was out there in the environment. Fortunately, I like this frowny face. Fortunately, there is a solution to this problem, which is to go to the environment, study organisms directly in the environment. And one of the most powerful ways to do that is to extract DNA from an environmental sample and look at the DNA from those environments. And what you can do is go to any environment, extract DNA from some environment, and read the sequence of the letters of the DNA from things from that environmental sample, just like Woese was reading the sequence from the organisms that he could grow in the lab. And then you can compare, I'm just showing you can do this with many organisms at once, you can compare the sequences to each other and place them into an evolutionary context. Sort of like if you got feathers from birds on an island, you could figure out what types of birds they might have come from in the same general sense. And this, being able to study organisms without growing them in the laboratory, completely revolutionized environmental microbiology in all sorts of different areas. Yeah?
So if you're collecting DNA out in the environment, how do you know all the DNA is coming from the same organism, or how do you separate out the different organisms?
Yeah, so you don't know it's coming from the same organism, and you hope in many cases that it isn't. So if you go to an environmental sample that has, say, 1,000 organisms in different species, which I've shown in these colored dots here, you get at the end a piece of DNA that corresponds to each of those different organisms. And you can separately characterize them at the same time from an environmental sample. So it's like getting a big pile of feathers from this island, and you can at once say that there are some warblers there, and some sparrows, and some hawks, and some owls.
You can immediately identify the fact that DNA is from different organisms.
You know, let's just pretend like the years of informatics, and we're going to pretend like it's easy for a few minutes.
Okay.
It's actually quite complicated and quite hard to do this well.
Okay.
But it works good enough that I can, talk about the results
Okay.
and with reasonable confidence. So one thing that I'm really interested in are organisms that are mutualistic symbionts of other organisms. So for example, a lot of these organisms from the bottom of the ocean that I was mentioning before that feed those tube worms, you can't grow them in the laboratory. Nobody knew anything really about them because of that. But with this method, you're able to figure out that many of them are closely related, for example, to E. coli, bacteria that lives in our gut. You can use a lot of the tools of studying E. coli to study those organisms based upon figuring out what type of organism they are. Here are, again, some of these. This is what I spent 1.5 years sequencing one little piece of DNA from one of these things. when I was an undergraduate. Another really important thing that this method showed was if you build an evolutionary tree of organisms, don't worry about the exact details here, and you ask, what are the different major groups? If you know the thing, kings play chess on fine-grained sand, kingdom, phylum, class, order, family, genus, species, you could say that each of these represent a phyla of bacteria, or a phylum of bacteria. And at the time, there were about 37 of these known when a friend of mine made this figure. Of those, more than half of them, no one had ever grown any representative of that entire group in the laboratory. So imagine entire lineages of organisms that we know nothing about because they've never been grown in the lab. That's what this method also told us. It told us what we didn't know, in addition to what we knew. It tells us biogeography, so we can map the presence and absence of organisms across space using this method by finding the same piece of DNA in different places, just like if you found, the hair from the Yeti or something like that in all sorts of different places, you might conclude where the Yeti's been running around or something to that effect. You can compare samples to each other even though none of them have ever been grown. And of course, one of the things that people are doing now with this method is going like gangbusters through the human ecosystem, the human body. There's a huge project funded by the NIH to do what's called the Human Microbiome Project, to survey the microbes that live in and on us. The diversity is immense. There are thousands of species inside your gut. There are maybe even more on your skin. And there's lots in your mouth, on your face, in your ears, in other places we're not going to talk about. And people have used this method to compare the microbes in these different places across different people in response to health, disease, diet, etc. I'm just going to show you a tiny bit of that. I've said this before, people have seen me talk about this. I don't know why they had to show a semi-anatomically correct figure in the paper, but I have censored it. So if you compare the microbes in the colon and classify them into those different major lineages, the phyla of bacteria, they're very different than the microbes on your skin, or in your mouth, or in the vagina, or in other places. Basically, you can view the human body as an ecosystem. I mean, every little space is going to be different, and we know very little. We're just starting to learn about the microbial diversity in these different places. This is an unbelievable study where they went to even more detail, and they surveyed like the right and left hand, the palm, fingers, different parts of the forehead, and found, there were some similarities, so skin tended to be similar to other skin samples as compared to the gut, but there are different niches, so the back of your elbow is different than, the front of your arm in what microbes are there. This poor person had his face drawn up and sampled in every position around his head. He was actually one of the students in the lab who did this, so I don't think he complained. People are surveying the microbes after birth to study the colonization of infants using these methods and learning about what determines how you get colonized after birth and what are good things and what are bad things. We're literally covered in microbes and microbial genes, and so this I stole from a PhD student's thesis, which was asking, should we redefine what people are? Because even though I might have 99.9% genetic similarity with other people, the microbes that are on me might be very different from those people. And that might sort of define us. There are more microbes on your body than there have been humans on the planet.
Is there any study that has at all looked at people who have been on long-term antibiotics?
Yeah, so I didn't include some of these studies, but there have been a couple of studies recently, less about long-term antibiotics than about pulse chase antibiotic treatment. So David Relman at Stanford has published a couple of papers on this now where he treated with Cipro in the initial study and then studied the response to the treatment and then whether or not they recovered back to normal. And what happened was in most of the people after the first treatment, they seemed to recover back to normal. And then he gave them another treatment and looked at the response, and some of the people never recovered back to sort of their center that they had been at before, but many of them did. So it seems like there may be in essence, the ecologists call it multiple stable states, that if you disturb an ecosystem in one direction, it may sometimes never go back to its original state. So again, you have to think of this as like a forest. And if you clear cut a forest, sometimes you get the same things coming back, and occasionally if things are just a little bit off, and you don't have seeds for something or whatever, a different thing came back. And there have been, 3 studies published, basically, I think. We know nothing still. So, but the Human Microbiome Project, I think they funded about 10 or so clinical trials on antibiotic treatment to try and do big surveys of large collections of people to try and get some handle on what happens. I don't know of any doing long-term antibiotic treatments. But it's, it's very early in this learning. What we're at now is that people realize that the DNA sequencing methods are really cheap, that the way to study these is through DNA sequencing, and now they're imagining what experiment can I do. Whereas, again, when I did my project, it was, a year and a half to count one microbe. And now with one of these Illumina machines, we can, for maybe $1 a sample at most, characterize 10,000 microbes in a sample. So you could survey a person every day for 20 years, or 200,000 people in response to some diet change. And that's just happening now, just starting, because the technology has changed in the last year and a half to allow it to be cheap enough that you can do big studies. That sort of makes sense? So basically people are arguing that you should view your microbes, the microbiome, as an organ in essence. Even to the extent that they are now doing, let's just call them microbial transplants, although the real name is fecal transplants. And they have cured people of things like C. diff infections with transplants of microbial communities from healthy individuals. I think it's probably too far a stretch to call the microbiome technically an organ, but it does all sorts of things that contribute to the biology of the host, with digestion, management of the immune system, killing pathogens, healing wounds, contributing to obesity, contributing to autoimmune disorders. We don't know in many cases what the cause and effect is here. There's still a we have correlations with changes in the microbial community and some health status.
Is there any possibility for gene regulation from the microbe?
You mean, can we regulate their genes or can they regulate our genes?
Yes, the latter.
The latter, yeah. So there are a lot of studies that have started to look at this in animal systems, and they have found that we've known this for pathogens for a long time. Pathogens do this routinely, manipulate the host in order to hide, or not unusual at all. The question is, do these commensal microbes do the same thing? And to me, it would just be completely ludicrous to think that they wouldn't.
Yeah.
They've been co-evolving with us for maybe even longer than many of the pathogens. And so now people are in fact discovering many of them are manipulating host hormonal signaling pathways. Gene expression has been shown to be changed, although it's not clear what the mechanism is. So people have done this in particular in what are called germ-free animals. You can raise mice with no microbes. Don't let them get colonized after birth and go through a few generations of complete cleanliness, and eventually they have no microbes. No microbes from the outside. Some of them have viruses that are hidden in their genomes and things like that, but no sort of external microbes. And then you can introduce a single microbe into them and study the gene expression response of that single microbe versus another single microbe. And so people are starting to get a handle on what microbes might be sort of the mechanisms by which they might be communicating with the host. Are they secreting small molecule compounds? Are they burrowing into nerves? Are they, it's not clear exactly what all of them are doing. But there's no doubt that they're going to be manipulating host biology just like the pathogens are. I don't know, I just have a couple more minutes. So now I haven't, this is from the Genome Center, I haven't told you about genomics because I wanted to tell you about this environmental stuff first. But the third era so the environmental stuff started in about 1985 was when this person, Norm Pace, first did this environmental RNA surveys from organisms in the environment. And then in 1995, a big change happened again in microbial studies when this place, Tiger, the Institute for Genomic Research, sequenced the first complete genome of an organism. So what I've been telling you about mostly has been people looking at one tiny region of the genome of organisms. One gene inside a sea of thousands. So an average bacteria will have 1 to 5,000 or so genes. And this one gene that everybody was looking at to do these environmental studies was particularly similar across lots of species, so they could compare bacteria to archaea to humans. But the functions that are encoded by microbes that are interesting and vary among organisms are in the rest of the genome. And so in 1995, the first complete genome sequence of any organism was determined at this genome sequencing institute called TIGER, and they used what's called the whole genome shotgun sequencing, which is what we still basically use to sequence genomes, where you extract the DNA from an organism, you blast it apart with your space modulator or some other technique. It breaks it apart into lots of little pieces, and you read the sequence of those little pieces. In the past, we used to think this was a really fancy machine because we could read 384 sequences at a time on that machine. Sad what has happened. It's sitting in the corner now crying. And then you take those sequences that you get out of that machine and you compare them on the computer to each other and look for overlaps between them, so that they came from the same region of the genome of that organism. And then you can stitch back together the genome sequence of the organism by doing that. And there's lots of bells and whistles. This doesn't always work perfectly, but eventually you can get a large sample, if not the complete sample, of the genome of an organism. And this revolutionized microbiology. I mean, it revolutionized lots of other things too, and that's why we have a genome center here. Microbiology has been sort of viewed as the golden age of microbiology now because of genomics. In part because the genomes of microbes are pretty small. So they're 1,000 times or more smaller than the human genome. So the cost to do a human genome, we could do 1,000 microbial genomes. Now there's lots more data from all sorts of different microbes while we're still sort of crawling along getting data from the organisms with large genomes. And don't really have time to go into all the detail, but it really did revolutionize a lot of approaches to microbiology by having the genomic data for an organism, which then you could catalyze experiments and learn how to, what was making an organism pathogenic, and do functional studies based upon the genome. You can predict all the metabolic processes in an organism. This is a picture we made for Vibrio cholera. They sequenced the genome of this organism, and I saw that paper, and I said, when I was a graduate student, I have to go to that place. And I dropped basically what I was doing and learned how to do genomics, and eventually moved to that place that did that sequencing. And I helped analyze the genome of Vibrio cholera, the causative agent of cholera. And even though this picture isn't perfect, we could do a pretty good job of predicting every transport pathway that this organism used to move things in and out of its cell, and every metabolic process in the organism. It wasn't perfect, but it was much more than we had any idea about previously. Another thing that's very vexing from genome analysis is that it turns out if you compare the genomes of 3 things that are said to be the same species, so 3 different strains of E. coli, turns out they can have thousands of genes different between them. So if you compare 2 people, we will have a lot of genetic similarity. We'll probably have almost all the same genes. How the genes are regulated will be different, the flavors of what genes we have will be different, but we won't vary that much in the presence and absence of genes. Yeah?
How can you tell how this organism is moving materials in and out from the genetic sequence?
Well, this doesn't tell you how it's moving them in and out, but if you compare the 3 genomes to each other, you can find that they share about...
No, this is the previous slide.
Oh, so this is... you do what's called annotation. So in the genome, you get the genome sequence of this organism. It's actually pretty easy in bacteria to predict where the genes are that code for proteins in the genome. And then you take the amino acid sequences of those proteins, and you compare them to the amino acid sequences of proteins of genes that have been experimentally characterized in other species. And it turns out that the things that move things in and out of the cell have been very well studied. And there's a great database of the different forms of transporter proteins and the type of things that they move in and out of the cell. And they classify pretty easily by the sequence of the protein. So the guy who designed this database sat next to me too. So he was at the same institute. And it works pretty well for that part of biological predictions. Other parts suck. I mean, just don't work very well at all. So we can make predictions. Some of those predictions are bad, some of them are good. But the people who studied Vibrio cholera, this was a revolution, to have that template to compare to instead of 20 experiments that have been done on those things.
Tremendous medical implications in knowing what kinds of materials they can move in.
Yeah, so Rita Colwell, who then became the head of the National Science Foundation, was our collaborator on this project. They then went and used the genome sequence to design studies to figure out what Vibrio was doing in the waterways to survive. So that was leverage from the genome sequence, and Rita has now gone ahead to sequence like 10,000 Vibrio genomes. I think that's what she told me the last time. So the genome really did revolutionize all sorts of aspects of microbiology by allowing you to make predictions, and if you had confidence in those predictions, then testing them with some experimental study. Just gonna tell maybe one and a half last stories here. So another thing that genomes allow people to do is get a better handle on the evolutionary relationships among organisms. So this ribosomal RNA study that Carl Woese did, and then the environmental studies that people did, could basically classify microorganisms in the big picture. It basically allow you the equivalent to, you could say that something was a warbler with birds. But you couldn't really tell the different flavors of a particular subtype. The gene that people were looking at did not change very rapidly over evolutionary time. Sort of the equivalent to saying the presence of, 5 finger bones or something like that. I mean, it allowed you to classify things at the big picture level, but not at the fine scale resolution. And so many people looked for other means to classify organisms in addition to this sort of ribosomal RNA data. And when I was at Tiger, the Institute for Genomic Research, we had previously sequenced the genome of the Ames strain of anthrax, which is the laboratory strain of anthrax. I was working on this project that people use to study the biology of anthrax. And then the anthrax letters happened. And the FBI and various other agencies came to Tiger and said, can you help us figure out where the anthrax in these letters came from by sequencing the genomes of those? And the reason they asked us that was because there was a way to classify anthrax without sequencing the genomes. This guy named Paul Keim had gone around and surveyed anthrax from all around the planet, collected samples from all around the planet, developed a method to classify anthrax by surveying portions of the genome of anthrax. And he could classify anthrax to some level of resolution with his method using that approach. And then when the letters happened, he used his approach to say that it looks like that this anthrax comes from a relative of this Ames strain that people were working with in scientific laboratories. But he couldn't say anything more. He couldn't say that it came from the Ames strain that they were working with in Iowa, as opposed to the Ames strain that people were working with in England. So the FBI came and said, can you read the entire genome sequence of these anthrax strains and maybe better classify where it came from? And that's actually what Tiger did. Colleagues of mine there then did analysis of this data. So the history was the Ames strain came from a dead cow, not actually in Ames, Iowa, but it was sent to Ames, Iowa soon afterwards. That strain then, people had historical records saying it was then sent around the world to all sorts of labs that then studied it. And then the question was, here's the attack strain in the letters. Is it more closely related to the anthrax from lab 3 or lab 2 or lab 1? In the time that people had been growing the anthrax strain in their labs, it should have accumulated mutations that would allow you to distinguish one strain from another strain from the labs. And in fact, that's exactly what they found. So they sequenced the full genome and then found that you could, not perfectly classify, but you could classify it at least at a finer level scale of resolution, and that assisted in the investigation to tell them where the anthrax in the letters may have come from. So this is, in essence, you could call it genomic epidemiology. That was the first example of this. It's now routine. So the E. coli strain in the Germany outbreak, the swine flu last year, I'm sure if there's any other flu epidemics, everybody is doing whole genome surveys to do sort of comparative studies of the epidemiology and the history of particular outbreaks. just like was done with anthrax. Okay, so in the last tiny little bit here, so I've told you about doing DNA studies of organisms you can grow in the lab by looking at this one gene, the ribosomal RNA. DNA studies of organisms in the environment by looking at ribosomal RNA in the environment. Genomic studies of organisms you can grow in the lab. The obvious next thing to do is to do genomic studies of organisms in the environment. And that's exactly what happened. So there's a new whole approach to studying organisms in the environment, just like genomics has revolutionized the study of microbes that we can grow in the lab. So-called metagenomics, taking samples from the environment and reading the entire genome sequence from those environmental samples, is revolutionizing what we understand about microbes in environmental samples. So it works in the same general approach. You go to an environment, extract DNA from the environmental samples, and you run it through this shotgun sequencing. And people are doing this now from every ecosystem you could imagine, from the human ecosystem, from oceans, from inside buildings, from deep sea hydrothermal vents, from borax lake, from anywhere you can imagine. People are now trying to read the entire genome sequence of organisms from the environment in addition to what they had been doing previously. I'm going to leave it there. I'm not going to tell you about all the findings that have come about it. Some of them are very exciting. I'd be happy to talk to people about this anytime. It is the new area of microbiology research, but it is what's going to allow us to get to this point of having a field guide to the microbes, which is my dream. And I will leave it there and say thanks to a lot of people who I've stolen slides from and pilfered money from. And thanks to all of you for coming here and listening.
Thank you very much.